Hello friends, Ace Botane next episode. In this episode, we'll be discussing NCERT quick reference. Last episode we done 8.1, 8.2 of Cell, the basic unit of life. So in this episode, with a minimum time, 8.3 and 8.4 will be discussed. So for convenience, I will share the screen so it will be easy for you. I think screen came, so we can proceed. So NCERT quick reference, because detailed notes already there in YouTube. So we'll be discussing an overview of cell and prokaryotic cell. 8.3, 8.4 edition is NCERT reprint edition March 2024. So we can start. So when we start with 8.3, an overview of cell. Onion peel cell is a typical plant cell. And there we can find a cell wall. Cell wall will be there. Inside it, there is cell membrane. Inside it, cell membrane. A typical plant cell. So cell wall will be there. Inside it, cell membrane will be there. Now when we come to a typical animal cell, that is human chick cell, here we can find no cell wall. So only cell membrane as outer layer. So outer layer is cell membrane. Outer layer is cell membrane. So plant cell, animal cell. I think that idea clear. Now in both cases, since we are considering it as a eukaryotic cell, so both case, plant cell, animal cell, typical plant cell, animal cell, there will be nucleus. Nucleus can be seen which contain or with the chromosome. Chromosomes will be there, and that contains genetic information. Genetic informations will be there. That is DNA. I think that aspect is clear. Now we can proceed to a typical eukaryotic cell. So some that I written there to save time. In typical eukaryotic cell, it is having membrane-bound nuclei. Here we can find cell organs. So cell organs also having membrane. In both prokaryote and eukaryote, we can find cytoplasm is present. Cytoplasm is present, and cytoplasm is the important statement, main area of cellular activities. Usual statement type question. Now, metabolism. Metabolism seen in all living cell. All living cell. That means keeping life activity. Keeping life activities. Life activities or maintaining living state. And various organelles we can find in a typical eukaryotic cell that include endoplasmic reticulum, Golgi body, lysosome, mitochondria, microbodies, vacuoles, etc. I think first page is clear. Now we can proceed to second page. Main content will be discussed. Now we have to proceed with the ribosome. Ribosome is the smallest structure. Famous question. Smallest structure, whether in eukaryote or prokaryote, smallest structure, and no membrane. So without membrane. Without membrane, seen in prokaryote and eukaryote. So ribosome is an important structure. It is concerned with the protein synthesis. And this will be the appearance of ribosome. The detailed study will be doing later. And already detailed discussion in their YouTube. Now, ribosomes. Which all area we can find ribosome? Ribosome is seen in cytoplasm, mitochondria, chloroplast. Now, in animal cells, we can find specifically a structure called centrosome. Centrosome. And remember, like ribosome, it is also membraneless. Centrosome also membraneless, amembranous. And main function is helps in cell division. Helps in cell division. Now, size and shapes of cell highly variable, and that is correlated with their functions. Now, some size given in NCERT, it's a database question, match the following diagram-based question. Mycoplasma, smallest cell. And NCERT statement, it is given as 0.3 μm in length. While in diagram, PPL law about 0.1 μm. So statement and figure. Data slightly varying. Now bacteria, it is 3 to 5 μm. Largest isolated cell is ostrich egg. Among multicell organisms, human RBC, 7 μm in diameter. Nerve cell, some of the longest cell. Size and shape of cells, highly variable. Again, size and shape of the cell, highly variable. Highly variable. Size and shape. And again, connected with the function. I think idea clear. Now we can proceed to next page. Yes. Now, prokaryotic cell. So prokaryote means no well-developed nucleus or cell organs with a membrane covering. So no membrane covered organelles. Typical example, you're familiar, bacteria, blue kidney algae, mycoplasma, yellow. And normally, these prokaryotes are generally smaller and multiplies quickly than eukaryotes. Shape and size, again, variable, prokaryotic cell. Then 4 basic shapes. Shapes important. It is bacillus, coccus, vibrio, spirillum. They are the main names. And the shape will be respectively rod, spherical, comma, and spiral. Organization, fundamentally similar. In all prokaryotes. Next is cell wall. Cell wall is seen except mycoplasma, where it is wall-less bacteria. Inside cell wall, cell membrane, cytoplasm, and no well-defined nucleus. Now, naked genetic material, genomic DNA, which is single chromosome, chromosome inverted comma, and it is having circular DNA. Now, many bacteria have small circular DNA located outside the genomic DNA. It is called plasmids. Important for exam. Theory entrance. Plasmids. It is small. And different types of plasmids are available. And plasmids, they are specific. And they have specific functions also. And different plasmids include R plasmid, F plasmid, cold plasmids. Details are given in detailed description. Prokaryotes also possess inclusions, non-living materials, inclusions seen. Then, infoldings of cell membrane, it is called mesosomes. They connect like mitochondria. Then, chromatophores, also seen in prokaryotes with the pigments. So, helping in photosynthesis. Okay, so that is regarding the second episode, cell 8.3, 8.4. If you like this episode, subscribe or like it, share it, comment on it. We can meet later. Meanwhile, shorts question I'm daily doing 2 each. Then detailed note link also will be provided. Okay, thank you. Take care. Bye. We can meet later.